happen if we do things differently here. I'm going to talk about two concrete examples. The first one is a little bit, it illustrates the principles, but it's obviously not a real problem. So imagine that someone did a randomized trial on the effects of Russian roulette in Russia. And they got some results, and they want to know whether those results also apply to Norway. Let's say in this setting, the experiment was that everyone in Russia had to do Russian roulette once a year. And we want to know the 1-year mortality, the effect on 1-year mortality of this intervention. And suppose what happened is that the mortality in Russia went from 1% to 17% over 1 year. Now imagine we want to extrapolate this to Norway. So if we are using the approach based on effect homogeneity and distribution, we're going to have to control for everything that is associated with death in Norway and Russia. Every cause of death that differs between Norway and Russia. In contrast, if we wanted to do something based on effect measures, we want to control for everything that is associated with the figure 1/6, which is the actual biological effect of putting a bullet into one of the 6 chambers of a revolver. It might be that Russians who play Russian roulette tend to be drunk and miss, so that they have a lower probability than 1/6 of being killed. If that is the case, we might need to control for alcohol use in order to do this extrapolation. But we do not, if we use effect homogeneity and measure, have to control for every cause of death that differs between the populations, only the things that determine the figure 1/6. But note also that 1/6 is something that comes out of the probability of survival, not the probability of death. If we use the probability of death, it would be 17/1. The normal risk ratio is 17/1, and it will not be stable between populations with different baseline risk. Whereas if we use the probability of survival, if we take 83 and divide it by 99, we're going to get 5/6, which is just the remaining probability from 1/6. Okay, so the correct effect measure here is not the effect measure that captures the idea that the figure 1/6 is equal between populations is not the normal risk ratio, but the survival ratio, which is a very simple biological example of the logic that underlies the framework that I'm trying to introduce. Okay, so to give you an example that is more applicable and gives you more an understanding of why I think this is genuinely important. Suppose we're looking at allergic reactions to a drug, and we're in a population where 1% of the population will have an allergic reaction no matter what. So 1% of the study population, the placebo arm, gets an allergic reaction. And 2% of the study population get an allergic reaction when they take the drug. Okay, so we have a risk ratio of 2 and a survival ratio of 98/99. Now suppose we go to a different population where people eat a lot more peanuts, so there's a lot more higher baseline risk of having an allergic reaction. So if the baseline risk is 10% in this peanut eating population, it would be insanity to think that they would have a doubling of their risk of an allergic reaction if they take the drug. Whereas if we think in terms of the survival ratio scale, it might be that 1% of the population have the gene that causes the drug to give an allergic reaction. So what you will see is approximately that 11% will get an allergic reaction if they take the drug in the peanut eating population. You now have a biological explanation of what it is that leads to the effect being equal, it's basically stability of the distribution of that gene that is associated with having the effect. So what I'm saying is that doubling is a concept that doesn't make any biological sense at all as something that could stay stable between two populations. And we should stop talking about a drug doubling the risk of an outcome. But it makes sense to talk about the prevalence of whatever it is that is causing the drug to have an effect as being stable. And that will often lead to a quality of the effect of introducing the drug and not to a quality of the effect of removing the drug from a fully treated population. Okay, so the idea that I'm talking about has arisen independently at least 5 times in the previous literature with subtle variations. The original paper by Mendel C. Sheps in the New England Journal of Medicine in 1958, Shall We Count the Living or the Dead?, is a classic with over 100 citations, but nobody listened to her, probably partly because of the difficulties in being a female statistician in the 50s. Paul Glasio and Les Ervig published in the BMJ in 1995 with a proposal that is almost the same if the outcome we're talking about is rare. What they suggested was to consider multiplicative effects for beneficial outcomes and additive effects for harmful outcomes. That is essentially the same as long as the outcome is rare. Dijk's paper in Statistics in Medicine I've already briefly talked about, which is an empirical suggestion that corresponds to my theoretical development. There are also 2 recent papers in the Journal of Clinical Epidemiology and on Archive that make essentially the same suggestion. So, in my view, it is time for people to actually start doing this and to follow the recommendations of the Cochrane Handbook and use the normal risk ratio for the probability of death for drugs that reduce the incidence, and when we talk about side effects, to use the survival ratio, the recoded risk ratio, the risk ratio for survival, as our measure of what the effect is, so that we can define homogeneity between populations in a way that makes sense in terms of a biological model. What I add to this discussion is that I provide a formal counterfactual causal model to explore the scope and limits of the underlying intuition. Okay. So my papers on this are available on Archive. The first paper, called The Choice of Effect Measure for Binary Outcomes, Introducing Counterfactual Outcome State Transition Parameters, has been published in the Journal of Epidemiologic Methods. The second paper, which introduces some weights for standardizing effects between populations, is at the revise and resubmit stage. And the third paper, which explains the differences between this approach to homogeneity and the one that's been suggested by Pearl and Barnboim and by Stewart and Cole, explaining the differences between those 2 ways of thinking, will be submitted to scientific journals within the next week. So thank you so much for listening, and please do send me your questions at anders@whitfeld.net. Thank you so much.